this is recording.
RTI
International
Center for
Forensic
Science
presents
Just Science.
Welcome to Just Science, a podcast for justice professionals and anyone interested in learning more about forensic science, innovative technology, current research, and actionable strategies to improve the criminal justice system. In episode 3 of the 2020 R&D season, Just Science interviews Dr. Carl Wolf from the Medical College of Virginia Hospitals about 40-plus ways not to analyze beverages for cannabinoids. From professional student to a leading researcher in the analysis of cannabinoids, Dr. Wolf has been involved in academia for decades. In that time, he has consulted and lectured on toxicology, given expert testimony, contributed to over 100 presentations and peer-reviewed publications, and worked on multiple NIJ-funded grants. Tune in as he talks about academia, analyzing beverages for cannabinoids, and the value of failure in this episode of Just Science. This season is funded by the National Institute of Justice's Forensic Technology Center of Excellence. Here is your host, Dr. Megan Grabenower.
Hello and welcome to Just Science. I'm your host, Dr. Megan Grabenower with the Forensic Technology Center of Excellence, a program of the National Institute of Justice. Today our guest is Dr. Carl Wolf from the Medical College of Virginia Commonwealth University. He's been on the Just Science podcast before in an episode called Just Live or Die, where he discussed his other NIJ-funded work about postmortem work in forensic toxicology. Carl, welcome to the podcast.
Good morning, Megan.
Good morning. So before we begin on your current NIJ-funded work, do you have any updates on your Live or Die presentation from?
Liver doesn't die, yes, and other things we learned. Well, that is actually after much prodding from students and fellows of mine. We submitted that last Friday for publication as a manuscript, so we'll see what comes out of that.
Oh, nice.
We ended up with 12 different opiate assays that we evaluated, commercially available ones, and our own in-house developed one. And the data was kind of interesting, as I presented at the meeting, and I've presented it a couple other times, is that none of them worked ideally for liver tissue. Surprise on that one, compared to urine or blood. But that if you understand the advantages and the disadvantages of each method, I think that most of them are usable in a postmortem liver, doing liver analysis. They don't work the same as blood and urine, but they will do what you need to get out of it.
Nice. Well, congratulations on getting the manuscript out the door. It always
Thank
feels
you.
good to put it in someone else's hands then.
Yes, it does.
I see from looking over your bio that education is a pretty big part of your career. Is it still a big component of your daily work?
At one time, I was a professional student for many years of my life. And then it was one of those, I want to say, circumstantial things that happened in my life. Things came up and I didn't get my degrees when I got my bachelor's and whatever, but my master's and then my PhD took a little longer than I had planned on. And then it was sort of like in that I had opportunities to take just a few extra classes, like 144 graduate credit hours of classes. So I've had the ability of taking the basic things that people take, but then I got to take a lot of odd and weird things, which became and have become very useful in all the things that I do in my life. In my world, I talk to physicians and nurses and pharmacists and attorneys and judges and residents, and so being able to speak on their level is a great advantage. Then they're more willing to listen to you as not just, you're the expert, yeah, tell me what's going on, but if you can put it in their terms of what they understand, it makes it more personable, I guess.
Did you take any particular classes that at the time seemed irrelevant but have turned out to be pretty useful knowledge to have in your back pocket?
No, maybe I'm too old or whatever, but I had, in high school, we worked in chemistry labs that didn't have hoods. And we opened bottles on the counter, and I know there's safety issues, and I understand, trust me, I understand all that. I've blown up enough and burned up enough stuff in my life. I do understand that. But some of that is the wow of that is what got me into that, is the fact that, you can watch things spontaneously combust, or you can, mix things together and make a new product, and you're like, this is kind of cool. And now a lot of that is done online, but you don't get to really see, you don't get to go, oh, what happens if I add a little bit more of this? Where does it go? Or if I don't put enough in? We only teach success. And in my life, failure is far more often than what people call success. It's taken me a long time to get to where I am, and I wouldn't give up any of the failures that have happened, because that's how we learn.
Yeah, I can tell you seem very comfortable with that. We'll talk about that in a minute.
Sure.
So we're here this week at the American Academy of Forensic Sciences annual meeting. You presented a presentation called 40 plus ways not to analyze beverages for cannabinoids, and that was part of the NIJ Forensic Science R&D Symposium. If listeners are interested in watching the archived recording of that presentation, it will be on the forensiccoe.org webpage or the landing page for this episode. Are there any other co-workers on this project that you'd like to acknowledge?
This would not have happened if it wasn't for our clinical laboratory science student, undergraduate student who we had work on this project, Heidi Brightman, because the university offers what they call UROP, which is Undergraduate Research Opportunities Program, UROP. And they basically give students a stipend for the summer to work in their discipline. So this can be art, this could be history, social science, anything, not just science. They get to work with somebody in the field working on a project and move beyond and get a better understanding of, one, how to do research in their field, but also, results that you can generate. And so I have a colleague, William Corazon, who's one of the faculty there, who I said, hey, do you have any students for the summer? I got a project. And so he sorts through and goes, who's interested, who's not. And Heidi was like, I would like to do something for the summer. So I was like, cool, we have a cool project. Justin Pokless, because he and I have been friends and collaborating for years on various projects, VCU and VCU Health and everyone else there that's sort of made this a possibility.
The title of your presentation, to me, is one of the more interesting ones I think I've ever seen in a program for a conference, is being 40 plus ways not to do something. You're really laying it out there that this presentation is going to have a lot of negative results, and that's something you don't see too often. But it is debated in the scientific community, is that we tend to not disseminate or talk about things that didn't work. So why is it you felt it was important to bring that up here at the symposium?
You can thank my student Heidi for this one, because when she was working on it, she's like, none of these work. And I'm like, oh, that's a perfect title. That's great. And being a professional student for many years, I've seen lots of things that work and don't work. And I guess I'm not one to reinvent the wheel. I think there's far too many things that we do in this world that we end up reinventing the wheel because we don't disseminate information very well, whether it's positive or negative. Master students work on projects all the time, and I'm involved with some of them and just all over the place where things don't work. And that's okay. But for your PhD, sorry, it has to work, and I understand that. That's a great and wonderful thing. But when people publish things, they go, oh, we have a method for doing this. Okay, but you didn't tell me what else you did to get to there. Or you said we did one or two things a little bit different, but no one tells you, what the effects are of that. Because if I'm doing method validation, which is now a big part of everything we do, part of that is also, can I tweak this one way or the other? What if I add a little more acid or a little more base, or I add a little more solvent, or, what effect does that have? Do I change the pH a little bit? What effect does that have on my results? And that's really the more important thing, because it talks about, how robust your assay is. What can you do with it and what you can't do with it. So, because this is a new field in the world of, we've been doing drug testing and biologicals for years. And, cannabinoids is a big thing. And in the food and beverage is becoming a very large part of the economy, but no one really knows what you're getting. Now people, I think, presume too much that the FDA/some governing body is overseeing everything that's going on out there. And to my perception, that's not the case.
So let's talk a little more detail about the goals of your project. Can you give us kind of in layman terms the big picture of what the purpose and the goals of this project you presented this week are?
Sure. This is a small part of a bigger project, as I said in the talk. The original premise was because this was before the Hemp Act, the Agriculture Improvement Act of 2018. My thought process on this was the fact that, what happens to soccer mom when she's bought CBD for her child who has epilepsy or something like that, and it works for the child. And she's driving down the road and the police stop her for whatever reason that happens, and they go, "What's in the car?" And it's like, "Oh, it's a CBD brownie." And the police at the time, not understanding the difference between marijuana, or maybe she even said a marijuana brownie, you send it to the lab, and the lab goes, "It's THC." Well, in Virginia, that's a Schedule 1, so that's a felony conviction right there, no matter how much is present in it. So I was like, I want to be on the defensive side on this one, because I'm going to go, so how do you store these things? How do you test for these things, both by the officer and by the laboratory? And how do you know, what's happened to this? And there was nothing out there, and I was like, this is going to become a big, major legal issue, and before it shows up on the 6 o'clock news or the morning news as this is the worst thing in the world and laboratories don't know what the heck they're doing, I wanted to, set the record straight from the beginning, not to go back and have to fix this later.
Okay, so you set out to develop methods for measuring how much of the different THC and other cannabinoids are present in things besides urine and blood and the plant itself, but the cookies and the brownies and
Correct.
and other
Beverages
edible products.
became a part when the Agriculture Improvement Act occurred, and suddenly it went from, at my local Whole's food store, of a little box on the counter that said CBD that was locked up, to suddenly displays and aisles.
Everywhere.
And it's everywhere. And I was like, this is going to be another problem.
Mm-hmm.
This is going to be a big concern. So why don't we go there?
Nice. And you managed to do it in one summer, the beverages portion?
Because we just did beverages.
Well, props to Heidi. She must have been very busy, because that's a lot of work in a short amount of time.
She worked in a lab before she went back to school. So that was the nice thing about having that.
Where are you in the bigger project? When did it start, and when do you expect it to end?
Funding officially started January 2018. And next month, we will be doing the 1-year stability study of our homemade brownies, gummies, and dark chocolate, 1-year stability of our cannabinoids. The last analysis for the acid portion of that will be finished in the middle of March. So I will then be writing up my final summary to send to NIJ, then working on the manuscript to be hopefully out the door before the end of June, when the funding officially runs out.
So in your presentation, which was specifically about the beverage analysis, you were looking at a way to compare different extraction methodologies and how to know one worked well and one didn't work well. And you used some terms. It's called matrix effect and process efficiency. Can you explain what those are for the listeners?
So matrix effect is, what is the matrix / whatever your compound of interest is in, how much effect does that have on your analysis? Does it improve your detection of your stuff, or does it interfere with it? Because in most things we do in the toxicology world, the compounds we're looking for are in minuscule decimal point percentages of what we're actually looking for. The needle in the haystack. So just that needle, how do you get rid of the whole haystack so you can only see the needle? And that's really matrix effect, is how much does that haystack affect your analysis? And then the recovery is, okay, how many needles were really in that haystack? So do we get them all out, or do we get most of that out? Theoretically, we should be able to get rid of all the matrix and recover all of our compound of interest, but we know in science that's not really a realistic possibility. So we work with what's acceptable. We accept ideally about plus or minus 25% effect on our matrix, and our process efficiency is how well do we extract and clean up everything. And we'd like that to be 100%, but we know that it can be, we go with 75 to 125% to give ourselves some leeway to say, hey, this works or doesn't work.
Okay.
That's ideally. In practicality, because we're working with things that we have never, or very few people have ever worked at, I use the philosophy, if you can get, in the end, get about 50% of what you're looking for, that's good. If you can't get half, try something else. There have been a few compounds we have tested in the past where 20% was the best. We were trying to do lipophilic compounds out of brain tissue, and that's not it. Getting fat out of fat is not an easy thing to do.
So how do you actually measure these things in the lab?
We have a UPLC triple quad system that we use for all of our other opiate and cannabinoid testing. That's how we're doing them. It's probably a little overkill because most of these are in high concentrations, so we're ending up diluting our samples in the end to some extent before we do the analysis. But this is what we have.
So you work with what you've got.
So as in we do in all labs, we work with what we have, and if we get the opportunity that somebody wants to loan us, or we have someone that we can collaborate with who has something else. The problem with cannabinoids are they're Schedule 1. So I can't just go, hey, can I send these over there for you to do the testing on? I'd love to, but besides federal, that's a university policy right there.
Yeah, research with controlled substances, that is a hindrance sometimes in who you can get to help you out and analyze and can receive the samples and work on them.
You can come to my world, but nothing can leave here.
In order to accurately measure the extraction efficiency of all these different extraction methods, you had to prepare your own beverages so you know exactly what's in there, right? You can't say how well you took something out when you don't know what you started with. So can you talk a little bit about how you prepared the beverages?
The great thing about the internet and students is that, I had Heidi, and that's part of the first thing we do is literature review. What's out there, what's published, what have people done? Because, as I say, a lot of these people publish and go, we did this. Okay, but what did you do that you didn't work? And maybe this is the best you got, but I'd really like to know how you got there, at least some summary of that. So serving size is all the important thing in the world of food. And so we have 5 and 10 milligrams of THC, which is California and all those issues, Alaska, Oregon, Washington State, Nevada now. We had a size of THC that we wanted. So the next question is, what are we putting that in? In the brownie world, it was like, okay, Duncan Hine brownie. And they had a size. It was like, cool. And Haribo and some other gummy makers had a size for that. And chocolate was Hershey's and Nestle's, and it's like, what do you consider a size? Beverages was like, okay, they come in all different things. And we went to the FDA and was like, okay, labeling of beverages. All right, and I was like, okay, so what are people selling and what are people buying? People buy their 12-ounce can of Coke, Pepsi, whatever the heck it is, Mountain Dew, Dr. Pepper. It's like, and things come in 12-ounce. And if you get a larger size, they still usually correlate it back to a 12-ounce serving. We were like, okay, that is what we have a basis that everyone can understand. The perception is this is a 12 ounce is a serving. So that was our 12-ounce serving, which is 354 milliliters. So now we have 5 milligrams and 354 milliliters of beverage, whether it's soft drinks or pop, brewed things like, because as the comment is now, it's in tea and coffee, cannabinoids, and people are making beer and wine. So we were like, okay, there's our 3 matrices: sugar, brewed, and fermented.
But how did you get the cannabinoids into the beverages? Because they are not typically soluble in things that are mostly water, and
That
a lot
is
of those
correct.
beverages are primarily water.
Yes, and thankfully they are primarily water. Does well for us. So we had to make an emulsion, and my student did various research of seeing, what's out there, what are people doing? Because as I was like looking at this, taking a pharmacy class, I understand emulsions and how they make compounding formulations, and I was like, so we're making mayonnaise, is what we're really making. A suspension. Well, we don't want a suspension. We want an emulsion. We want it to go and stay is to the best. We're making salad dressing, only creamy Italian, not just Italian dressing. So what are people using out there to get the cannabinoids? You have to get that micelle partition. So you got to have the basis to understand where you're going. So fruit pectin seemed to be what a lot of people were using, and people use fruit pectin all the time for canning. However, finding fruit pectin nowadays is not the easiest thing to do. It's only got a few spots on a shelf because people buy pre-packaged. And I was like, we can buy a box of Sure Gel or something like that, but that's powder. That's too hard. We have to weigh that. But there's a liquid form. And I was like, liquid's excellent because we're working with liquids. So we got that. We tried some variations on that, and was like the one that was online, and it's on the presentation, the blog site, was like it seemed to work the best that we saw, just mixing them up and getting it to stay as an emulsion for the longest time so we could do sampling out of it. Granted, as we left it sit, it separated. But for at least for 15, 20 minutes or so, half an hour, I think maybe was the longest, we got a solution that was hopefully homogeneous. That's where that came from, was just trial and error. And we just used their proportions.
So do you know, is that how the commercially available beverages are prepared? Are they emulsions?
Yes, they are. When I was in Seattle doing the liver talk, that was, I went looking at the dispensaries because we were in Seattle, and what else do you
Why
do
not?
besides, yes, trying the other things? And I was standing there looking at some of the beverages in the aisles, and the one gentleman came up to me who worked there and said, "I've noticed you've been standing here a while." I was like, "Yeah." And I explained to him who I was, and he goes, "Oh, that's really cool." I said, "I'm just looking at your products and how these," and I was like, "That's a suspension." And he's like, "Yeah, you have to shake them up to drink them." And I'm like, "Oh, okay, that makes sense."
Interesting. All right, so you had to buy the cannabinoids, make the emulsions, add them to your beverages. So I know from my own experiences that buying cannabinoid standards can get pretty expensive pretty fast. Did you incorporate that into your experimental design or any strategies to kind of keep your costs down?
Yes. So that was the original thought process. We put money in the grant because standards we knew were going to be the most expensive thing for us to get. And that's a big part of our process. How we worked that in there, one of the original thoughts was, some of this stuff we could buy online, like CBD. Well, CBD was not available when we first started this online. Or in any of the stores. They said hemp oil, but then there was a little disclaimer that this contains no phytocannabinoids. So we're like, that's worthless.
Not going to help.
And you could get a number of things, but it's gotten, I will say in the last 6 months or a year, it's gotten a whole lot easier to buy stuff, but we're on the back side, not on the front side. So we were purchasing them as certified standards. That's still an issue of buying any of these in labs trying to do this, is how do you get enough reference material, not just to make your calibrators, but to make your control materials too. Because most people go, "Oh, I want to make enough that's going to last me 6 months or a year." Well, what has come with that is that we realize that we have serving sizes and we have sampling sizes. So we want to make our sampling, our proportion of our control, not much bigger than our sampling size. So we can freeze them / whatever, store them, and that's part of the stability process going on, is what's the best way to store these things. With beverages, we ran into the issue of cannabinoids are not very stable in beverages. Unless somebody knows something, or maybe it's the way we prepared them, whatever, but our high sugar content products, cannabinoids in less than a week were not really detectable out of the 9 we looked at.
Wow.
And the alcohol in the fermented lasted at least up to about 10 days. We didn't do any testing beyond that because the summer was over. And we still have some in the refrigerator, and maybe at some point in time, Heidi will be able to come back as part of her spring semester if she's got a couple days, and we'll run a batch of those and see how they look 6 months later. But we noticed that right away because we would make a batch to do our effects, to look at the effects, and she would be like, "This isn't working." And I'm like, "Did you make it fresh?" She goes, "No, I made a big batch, and we run it for a couple days." And it's like, it's not working.
So you had to make fresh batches each day for extractions, or
for
every few
doing the matrix effect and process efficiency, yes. When we did our method validations, we did the calibrators fresh every day, and then our control materials were run as stability issues. We ran those, we made them up and stuck them in the refrigerator, because nobody really freezes these things. And we left them out on the counter, and we didn't see much difference between that and refrigerated. So it was like, okay, just keep them in the refrigerator.
All right, so how did it go? I mean, you tried 40 things that didn't work. Did you ever find anything that did work?
Yes. Well, we found 2 things that worked. One, there's a paper out there published on doing it by methanol, and just doing a methanol dilution and running it. And that's why we do this, is to look, not to debunk people's things, but to go, okay, how is ours better? Because you're never going to get a paper published unless you're better than someone else.
Can't
That's
just keep doing
an
the same thing.
unspoken rule. So, we did methanol. And what we saw was our process efficiency on this, because we're just diluting them, was about 20% over all 3 matrices we looked at, which I go,
So
50,
that means you were only able to get out 20% of what you know you put
or
in.
the effect, the appearance of, yes, it appears that you only got about 20% of your compound out.
Okay.
So whether that's that you had so much suppression of it from the signal that maybe you got 100%, but you got so much suppression that you didn't get to see it, or you truly only got 20% out. That was an issue, but we were like, okay, if that's the best we can do, then that's what we have. The problem we ran into is when we were doing fruit drinks is methanol dilution doesn't get rid of all the pulp and all the other crap in there. And it doesn't centrifuge out either. So now you got to go to another step and you got to filter it, or you got to run it on your instrument and hope that you don't plug it up. And I've already plugged my instrument enough times over the years to go, that's not going on my instrument. And now we're doing a two-step process. So now we're diluting and filtering. So why don't we just do that together? Keep it simple. And for laboratories doing this testing, if it's a 10-step process, I don't see most forensics, government, whatever, private labs going, we're not going to spend all day to do an analysis. We want something that's simple and quick to do sample prep, throw it on the instrument, and if it takes 5 minutes, 2 minutes, or an hour to run the samples, so what? We're going home. We'll pull them off the next day, and we'll crunch the data and get it. So that's mine, is how fast can we work on it, but get a method that works. Really, what we found that was worked, because we had them from a project from 8 or 10 years ago, was the United Chemical Technologies FAST column. It's proprietary, so they won't tell you what's in it, but they have what we call a green and a black column. That's the labeling on the tubes.
So that's all you know, the green one or the black one?
The green one we know is for acid neutral extracts, and the black one is for basic neutral extracts. So most people use the black column because most drugs are bases. And we compared those in an initial work on this and doing gummies. And they worked really well. They had similar comparisons when we did sugar content, things in high sugar content. We ran them on our chemistry analyzers, and it's like, ooh, the sugar's gone. Awesome, this is great. So we had some previous experience with it, and my one student at the time presented it at the academy meeting, and it worked well. And the only difference we saw between the two columns was the green column did not take out our red food coloring, whereas the black one did. And I'm like, okay, so if there's color left, then something's not taken out of it. When we looked at it for doing beverages, we didn't see any difference with color, but what we saw a big difference was sugar content. So sugar is the hardest of the beverages to analyze of the three. It has the most matrix effect, and it has the most ion suppression, both ways. It enhances your signal in some cases, and it depresses your signal in other cases, and compared to the other two. And as I'm looking at this, as we did with the other edibles, is when samples show up in your laboratory, you don't know right off the bat what it is.
Right, it doesn't come with a nice little tag that says exactly what you're analyzing.
So when we were doing the edibles portion, my student was like, well, let's do this and this and this. And I said, no, they all have to work together. I said, because what do you do with the chocolate chip cookie? I said, better yet, what do you do with a chocolate chip cookie with gummy worms in it? You can't pick out the parts and then figure out which one has the cannabinoids in it. The law is written as total content. So you got to grind the whole thing up and analyze a portion of that. Beverages, people put sugar in their tea and in their coffee. You don't get to choose what's in there, and you can even have Irish coffee or whatever you want to call it, So you end up with this mixture of all three common matrices that you have to run into. So your method had better work for all three of them, or it's not really useful.
How many individual methods do you think it's realistic for an analytical lab that's going to go into business, or a publicly funded lab that needs to be analyzing these edible products? How many different methods do you think they'd be willing to implement for this analysis? Do you think they would, if it's more than one, they're not going to do it? Or do you think they'd have one for solids, one for liquids?
I think that's what you're going to have to do. Because when you do in the biological world, you still have to do some kind of sample preparation on your liver tissue before you can add it to your urine and blood test. So, that's still two tests, unless there's something else coming up. Yes, if you want to do food, and we'll just use food as a general thing, because we don't have to deal with gases too much in food, the original elements, solids and liquids, you're going to need to have two different methods because they don't act the same.
So in the results that you presented, it was fairly common, even the extraction methods that didn't work out so well. Yeah, your brewed and fermented beverages seemed to track pretty close, like they
Yes.
had similar performance, and then the high sugar ones were either way better or worse than that. What is it about those high sugar things do you think that might be interfering or causing that
Percent sugar
discrepancy?
content. I think because sugar is such a big molecule and sugar is such a large portion of this, we didn't do anything like distilled spirits for our alcohol. We were doing what's classified as beer, so things between about 3.5 to maybe 7 or 8% alcohol. We did a couple wines too, so we were in the 12 to 15%. Brewed are, it's whatever tannins and slash whatever is coming out of the coffee bean or whatever your plant material is. So the content is not that high. It's probably, I don't remember the amounts, but it's 90-some% water with flavoring / colorings in it. Alcohol, you kind of have the same kind of thing. You're in the 90%, 80-something with your wines, that it's water. With sugar content foods, the sugar is a high proportion of, read the label, is a high proportion, and sugar is a big molecule too, compared to small molecules. So it has a large effect on what's going on. We ran a couple sugar-free things too, and we saw sort of in the middle. So it's probably, some of those are fairly large molecules when you think about it compared to something like an ethanol molecule.
So things that had artificial sweeteners
Artificial
instead of
sweeteners
sugars?
too.
Okay.
Yeah. We didn't see as bad an effect, but it wasn't as good, or it wasn't as consistent, I want to say, with, say, the brewed and the fermented, somewhere in the middle. So that was like, okay, then that was better going, look, let's just deal with true sugary drinks. And some of those were generic and some of those were brand names. We looked at about 12 or 15 different things on the market and made our own concoctions in them and saw some that worked better than others. And that's my thing, is I think it's just the fact that you're dealing with just the pure size of trying to clean up that much of whatever it is in there and get rid of it.
So what's next for your research? You mentioned you've got some final 1-year stability time points coming up, but what's the next phase for you?
My funding ends in June. This summer, hopefully, I might have 3 students coming to work on emerging opioids and fentanyl analogs. So the CDC commissioned Cerilliant and Cayman Chemical to make opioid and emerging fentanyl analogs. Maybe this is my perception, but I thought we were supposed to get a method along with the standards. Maybe I read something wrong a long time ago, and that's, like I say, my perception. But I have the standards, but I have no real method, and no one's really published anything other than, a case where they said, oh, we did this or that by whatever method they used. So we have no way to screen for 220 some compounds and really see if what's going on out there. So what I have a couple students this summer come work on is that, and we're going to develop an LCMS method for screening, because quantitation of these in a person it's there and you're dead. Wow, that's not that's not a big leap of faith anywhere, especially if it's the only thing or one of a few things present in there. Wow, that's pretty simple. But how can we screen for these? So initially we need to literally infuse these and figure out how to chromatographically separate them, because a lot of them are isomers of each other or isobars, and you're going like mass spec, a simple molecular ion is not going to do this. Like I said, I have a couple students working on this, and this is our summer project, is to come up with I'm going to say hopefully a method, but I'll be really surprised.
Wow, you're ambitious.
Shoot high and then drop on back. But maybe 2 or 3 different things, or an initial, hey, this is how we can screen for all these. We may not be able to distinguish X, Y, and Z compounds, but if there's something there that elutes at that time period and has this similar molecular fragmentation, that it's one of 2 or 3 things. And then you can go, okay, Now let's take that, because it's a screening technique, and push it and figure out which one it is, if you care. So that's the process. And where it's becoming important is, this is funny, coincidence things, our med tox folks and the ER department guys said, hey, we need somebody to do testing for new fentanyl analogs out there on the market, or not on the market, that are happening, because we're a level 1 trauma center. And being from Richmond, you're on 95, so everything from Miami to Boston stops in Richmond at some point along the way. What they're seeing in their patients is that typically a patient on an opioid gets a shot of Narcan and you wake up. And heroin has been very big in Richmond for years. And then in September of 2016, fentanyl started getting cut into our heroin. And what they've been seeing with this is not just fentanyl, but fentanyl analogs is, so patients get a shot of Narcan or whatever in the field. They kind of wake up, they bring them in, they're out again, they give them another shot, they wake up. And if it's just heroin, that's usually enough for the patient, then observe them for several hours, and then they come back and they're cognizant and they release them. But what they're seeing now with patients is they come in, or they get a shot of Narcan or two in the field, they give them a couple shots in the ED, and sometimes they even have to hook them up to a Narcan drip, and then they get admitted to the hospital, and now you're dealing with patients that they're not responding as quickly as they would with heroin.
It takes a lot more resources.
And it takes a lot more resources and time, and so they're like, can we figure out a way to test for this? And maybe we can come up with a scenario of, boom, if the patient comes in and you give them a couple shots of Narcan and they don't wake up, or they kind of do, sort of a retrospective back look and go, okay, these patients need admitted to a ward/unit, and they need to be maybe on Narcan, or maybe they find out that something else works better afterwards as to treatment protocols for these patients. And I said, oh, we got these 200 and some standards. This would be cool. We could work together on this, because they're like, we don't want a quantitation. We just want to know, is it there or not? And I said, perfect. This is what I want to work on also. So we're waiting and working on IRB approval. I need to have a method. So that's the new research project.
So how convenient, when your funding runs out, you'll have time to work on that.
Yes, exactly. Well,
there's always a silver lining.
There is, and sometimes people step back, take a breath, not a bad thing, and go, okay, so where do I need to go now? Because obviously straight ahead is not going to work.
So that's all we have time for today. I'd like to thank our guest, Dr. Carl Wolf, for sitting down with Just Science to discuss his NIJ-funded grant. So thank you, Dr. Wolf.
You're welcome. It's my pleasure.
I'd also like to thank you, the listener, for tuning in today. If you enjoyed today's conversation, be sure to like and follow Just Science on your podcast platform of choice. For more information on today's topic and resources in the forensic field, visit forensiccoe.org. I'm Megan Grabinauer, and this has been another episode of Just Science.
In the next episode, Just Science interviews Dr. Tatiana Trejos from West Virginia University about the rapid detection of organic and inorganic firearm discharge using laser-induced breakdown spectroscopy. Opinions or points of views expressed in this podcast represent a consensus of the authors and do not necessarily represent the official position or policies of its funding.